Hello, my name is Çalışkan, and this is my friend Murat Işık. We are from Turkey, and we are here to present our project named Prediction of SARS-CoV-2 Mutations that Increase Contagiousness. The COVID-19 pandemic, which appeared in 2019 and has taken 223 countries under influence, has caused over 2.5 million deaths and over 110 million cases. The COVID-19 pandemic has reached this level of contagiousness because of the quick spreading of advantageous SARS-CoV-2 mutations.
Advantageous variants have occurred especially by mutations composed of change in a single amino acid. These mutations, just as they can cause changes in the structure of the virus, can also affect the efficiency of the interaction between SARS-CoV-2 and ACE2, which is a protein that the virus uses as the first step to enter the human cells.
In order to solve this coronavirus problem that deeply affects the whole world and causes this presentation to be done online, by feeding the experimental and literature knowledge of biochemical changes caused by the binding and S protein stability data to different machine learning algorithms, we have aimed to detect the SARS-CoV-2 mutations that have an advantage similar to N501Y and E484K mutations. Firstly, we collected the mutation data that inspects the SARS-CoV-2 and its target protein ACE2's interactions from scientific literature. Then, we processed the amino acid change features in the SARS-CoV-2 mutations. Using the k-means and expectation maximization algorithms in VECA, we aim to predict which SARS-CoV-2 mutations have the same effect as N501Y and E484K mutations. We evaluated these mutations that were grouped together with these two mutations in the scope of SARS-CoV-2 and ACE2 interaction. As a result of the analysis of the clusters obtained from the k-means and expectation maximization algorithms, it is seen that there are common mutations in the list of 5 most dangerous mutations. Evaluating the dangerous mutations from both algorithms, we have found these 6 mutations to be the definitive list of mutations with the highest possibility of being dangerous: N501M, N501W, Q414A, N354K, Q498H, and 460K. In the image on the screen, you can see that the 498 and 501st positions are on the interaction surface.
Among the 6 dangerous mutations, position 501 is repeated. So we emphasize that mutations in this position should be given special attention as the most important result of our project. In our project, where we aim to find the dangerous mutations of the SARS-CoV-2 virus, which causes the COVID-19 pandemic that the whole world is fighting, using machine learning algorithms. Using the experimental dataset we obtained from the literature, we introduced the dataset which contains mutations that improve or does not affect binding, contains 8 amino acid properties that we have created with our own Python code. To various machine learning algorithms, we determined the k-means and expectation maximization algorithms as the algorithms that give the most consistent results. As a result of our analysis using these 2 algorithms, we found that the optimal value for each algorithm to divide the dataset into is 10 clusters. And we determined the cluster containing the N501A and A484K mutations together as the set of dangerous mutations. When we examined the cluster of dangerous mutations produced by the 2 algorithms, we saw that there were common mutations in the top 5 most stable mutation lists. When we examined these mutations, we consensusly identified 6 mutations as those with high probability of dangerous effects on the spread of the coronavirus. As the 501st position is repeated in our list and is now one of the most common variants, we emphasize that the mutations in this position should be paid attention to as an important result of our project.
Here, you can see the resources we used in this project. Thank you for listening to us.